Hey guys, my name is Didi Jimenez, and today we're going to be going over ion channels and neuronal communication. We'll be going over what ion channels are, the types of ion channels, which are voltage gated, ligand gated, and mechanosensitive ions, and looking deeper into those channels. And we are also going to go over neuronal communication and how it occurs with the synapses and the two neurons, the receiving and the giving. Okay, let's dig in. Pore-forming proteins, also called ion channels, are what facilitate the flow of ions across membranes, be it plasma membrane or the membrane of intracellular membranes. Gating these ion channels, well, most of them at least, is voltage, but certain ones like K and Cl channels, TRP channels, ryanodine receptors, and IP3 receptors are voltage insensitive and are instead gated by second messengers and other intracellular and or extracellular mediators. Now that we have gone over what ion channels are, let's go over the types. There are voltage-gated, ligand-gated, and mechanosensitive ion channels. Existing virtually in all cells, the major role of these membrane proteins is to regulate cellular excitability. Ion channels are of crucial importance physiologically and virtually. Now, depending on the stimulus that they are responding to, division of ion channels occurs accordingly into three superfamilies: voltage-gated, ligand-gated, and mechanosensitive, which are a type of ions that I mentioned. And we will be digging deeper into these ion channels on the following slides. Okay? Let's first begin with voltage-gated ion channels, which there are four. There are voltage-gated sodium, voltage-gated calcium, voltage-gated potassium, and voltage-gated chlorine. So when perturbations in cell membrane potential occur, there is a voltage-gated response and a dependability on the selectivity for specific ions like sodium, potassium, calcium, and chlorine, which I just mentioned. These specific ion channels can be further subdivided into families depending on their major permeant ion. Let's look at them deeper. First, we'll start off with the voltage-gated sodium channels, which have the responsibility for generating action potentials over long periods of time. With this being said, this responsibility makes them targets of local anesthetics like lidocaine and benzocaine. On the other hand, we have voltage-gated calcium channels, which regulate intracellular calcium concentrations. With these ion channels doing this, they are responsible for a lot of different biochemical processes within cells. The release of neurotransmitters at synapse is probably the most important process that is regulated by these channel types. Also, heart diseases all the way to anxiety disorders are treated with calcium channel blockers, so they are very essential. Next, we have voltage-gated potassium ions, that is K+. These ions consist of the largest, most different class of voltage-gated ion channels. They are imperative in generating the resting membrane potential, but that's about it. Finally, to close the voltage-gated ions, we have chlorine ions, or Cl-, which are presented in vast amounts as they are found in every type of neuron. These ion channels are in charge of regulating excitability and cell volume. Not only this type of ion channels are known to add to the resting membrane potential as well. Our second type of ion channels are our ligand-gated ion channels. We'll be going over the anesthetics, antipsychotics, and antidepressant side effects, and it is divided into three families: the cis-loop ligand-gated ion channels, the ionotropic glutamate receptors, and P2X receptors. Well, like we mentioned before, many drugs like anesthetics, antipsychotics, and antidepressants target ligand-gated ion channels. This ion channel is named accordingly as they respond to ligand and divide into three families depending on their molecular biology and protein structure. Cis-loop is the largest family of all the ligand-gated ion channels. It is made up of four different receptors: nicotinic acetylcholine receptors, which are NACHR, are gamma aminobutyric acid, or GABA, receptors, are 5-hydroxytryptamine 3, or 5-HT3 receptors, and are glycine receptors. Out of these four receptors, the ones that are excitatory are NACHR. I'll highlight it over. Oh, I don't have it over here. And are glycine receptors. No, the excitatory ones are the NACHR and the 5-HT3R. And the other two are the GABA and glycine receptors that are inhibitory. Pardon me. Digging in deeper to these receptors of the cis loop, LGIC, NACHRs consist of the neurotransmitter acetylcholine, activating them endogenously, that of which many cognitive processes like learning, memory, and attention require. Not only this, the neurotransmitter also plays an important role in chemical signaling at the neuromuscular junction. Next, we have 5-HT3 and its activation. These receptors are involved in many sympathetic and parasympathetic sensory functions, depending on the system that this receptor is being involved in. The function is different in the central nervous system. It is involved in emesis, cognition, and anxiety. Adding on to this, it also helps regulate numerous cognitive, physiological, emotional, and metabolic systems. It also affects physiological processes like sleep patterns, circadian rhythms, appetite, aggression levels, and body temperature. Following on to this with GABA receptors, which are still part of the cis loop. These receptors selectively conduct chlorine through its pores. This leads to hyperpolarization, which results in an increase in the threshold for generation of action potential. Neuronal inhibition is in turn caused by the reduction in the probability of action potential generation. These receptors obstruct the wake-promoting regions and therefore have to do with supporting sleep. Adding on to this is anxiety disorder, which are a result of GABA signaling being impaired. Thankfully, unwanted brain excitability can be reduced with the benzodiazepines because they help in increasing GABA signaling in the brain, so it can be used to treat anxiety disorders. The last receptor that we're going over in this slide is going to be for the cis loop are the glycine receptors, which in the spinal cord, brain, and other areas of the central nervous system mediate synaptic inhibition. These receptors regulate the excitability of motor and afferent sensory neurons. These include pain fibers. Not only this, glycine receptors play an important role in processing both visual and auditory signals. With this being said, these receptors show to be useful in the development of therapeutic compounds. So these are important for therapeutic compounds. Well, a certain one of them. I think it's taking me longer to write than it is to talk. Jesus. Okay. And we'll go over these two in the next slide. Now, all the information before was just for the cis loop ligand-gated ion channels. So we're going to go over the other two families, which are the ionotropic glutamate receptors and P2X receptors. Activation of ionotropic glutamate receptors occurs by the neurotransmitter glutamate, hence the name, right? These receptors mediate the most fast excitatory transmission in the CNS, central nervous system. These receptors are also subdivided. In this case, the subcategories are alpha amino 3 hydroxy 5 methylisozole 4 propionic acid, well, that was a lot, or AMPA receptors, kainate receptors, and N-methyl D-aspartate NMDA receptors. Excitatory synaptic transmission at the majority of central synapses is mediated fast by AMPA receptors, that is, our alpha-3 hydroxy 5 methylisozole 4 propionic acid receptors. Neuronal excitability is regulated by our kainate receptors, while synaptic plasticity has to do with our NMDA receptors. To close off our ligand-gated ion channels, we have our P2X receptors. These are the most recently discovered receptors. They're found to be permeable in sodium, potassium, and calcium, and are activated by ATP. Expression of these receptors occurs in various tissues and have demonstrated that they play an important role in different physiological processes like nerve transmission, pain sensation, and various inflammatory responses. In the picture, we see a neurotransmitter already binded right here. It's already binded on there, which helps stimulate the opening once the ligand binds, which in turn activates a small G protein on the intracellular side of the postsynaptic membrane, which we'll go over a little more in our neuronal communication portion of the presentation. But yeah, that's it for our ligand-gated ion channels. The last of our ion channels is the mechanosensitive ion channels, and we will be going over the mechanical forces on the cell membrane, the detection and transduction of external mechanical forces. When an ion channel responds to changes in mechanical forces on the cell membrane, that is when the mechanosensitive ion channels come into play. The detection and transduction of external mechanical forces into electrical and chemical intracellular signals are the main role of these channels. These ion channels are involved in regulating blood pressure, cell volume, stimulation of muscle and bone development, as well as the sense of hearing and touch, therefore, response to pressure. Various conditions like cardiac arrhythmias, muscle dystrophy, neuronal degeneration, polycystic kidney disease, and tumor metastasis have all shown the involvement of mechanosensitive ion channels. In our picture, you can see that these ion channels are activated in the response to membrane tension, right here as it says pressure, which alters whether the conformation is open. We have open with the pressure and closed without pressure, whether it's open or in a closed state. Well, that one was pretty simple. That is it for ion channels. But it is important to remember that ion channels play a big role in regulating major physiological functions, so it is important to know their mechanism at a molecular level. We are now going to talk about neuronal communication. Communication of nerve cells occurs through a combination of electrical and chemical signals. Within the neuron, charged particles drive electrical signals in order to permit the rapid conduction from one end of the cell to the other. The four simplified steps of neural communication are the reception of the signal by the sense organs, neurotransmission to and from a neuron to another neuron or muscle gland, integration of information from the signal, and the action or response to the generated stimuli. But let's dig into the meat of this topic and go over them one by one. Our first thing is going to be neuron-to-neuron communication. We're going to talk about rapid communication and the structure. In order for the brain to have the ability of rapid communication, there are cells within it called neurons, or excitable cells, that allow it. The shape of a neuron is unique because, like most cells, they have the cell body, or the stoma, but the different aspect is that they have specialized thin branches called dendrites and axons. Chemical input information is received by neurons from other neurons through the dendrites and through the axons. They communicate information to other cells. Interestingly enough, our ion channel chat from earlier plays a role in neuronal communication. These proteins are abundant within a cell and allow the passing of small charged atoms from one side of the membrane to another, which is vital for communication. In the picture, we can see the dendrites on the neuron. Let's see if it lets me this time. We'll do red. The dendrites on the neuron, which are the receiving. They receive the chemical input. You can also see the soma here and the axon terminal down here, which is the other important part, the axon terminal, which is where the neuron communicates information to other cells. So this is output. Info. Sorry, it's taking me so long. This is hard. Input. output. Okay. This picture also shows you voltage-gated sodium channels located in the membrane of the sodium, which is what we talked about earlier. So I'll change the color because this has to do with our ion channels from earlier. These allow for positively charged sodium ions to enter the neuron, resulting in the production of rapid conduction of the excitatory action potential to the terminal. This type of signaling causes a neurotransmitter to release at the axon terminal. So these allow for charged sodium ions to enter. And there you go. That's a little rapid picture all in one. And now we'll move on to the next one. Our next thing is chemical interneuronal communication. And we'll be talking about the synapse, how communication occurs, and the structure. This type of communication involves the release of a neurotransmitter from one neuron, altering the activity of the receiving neuron. The area where this communication happens is called the synapse. That is here. Or I should have changed the color back to red, but it's okay. Synapse. Which is where the two cells are about 20 to 50 nanometers. Away from each other. The two neurons that are coming into contact also have their name. One is the presynaptic neuron, which is the one releasing the chemical. Releasing. This presynaptic neuron has an axon terminal at its tip, which has a small pack of vesicles filled with neurotransmitter molecules. That's this part here. We'll do yellow. This part here. And it has a neurotransmitter molecules. Once an axon terminal is reached by an action potential, it is stimulated. To rise the concentration of calcium, which the ion then stimulates, the fusion of the vesicle within the cell membrane, resulting in the release of the neurotransmitter into the small synaptic gap between cells. The acted on neuron is the postsynaptic neuron. This one is acted on. The neurotransmitter molecules released from the presynaptic vesicles traverse the synaptic gap and bind to proteins, termed neurotransmitter receptors, on the surface membrane of the postsynaptic neuron. So they go from here that way. In this picture, we can see the synapse between two neurons. The neurotransmitter is inside the synaptic vesicle, and it is released when the membranes fuse with the outer cell membrane. In order to bind to the receptor, both ligand-gated ion channels and G-protein coupled receptors located on the postsynaptic neuron, neurotransmitters must cross the synaptic cleft. GPCRs on the presynaptic neuron's axon terminal alter the function of voltage-gated ion channels and modulate neuron transmitter release. Neurotransmitter transporters remove neurotransmitter molecules from the synaptic cleft so that they can be repackaged into vesicles. Following that, we have our neuron transmitter receptors, which are two major classes. It's the ligand-gated ion channel receptors, which we mentioned earlier, and G-protein coupled receptors. Specializing in the direct chemical signaling of the neurotransmitter into an electrical response is the responsibility of our ligand-gated ion channel receptors. The part that specifically specializes in the binding to the neurotransmitter part is the binding site, which is located on the extracellular part of the protein. Binding. Let me write that. So the binding. That says binding, by the way. Binding. The part of the protein buried within the cell surface membrane forms an ion pore, which is a fluid-filled hole in the membrane through which the charged ions can pass. And here we have the pore, which ions go through. Keep in mind that this whole opening and binding mechanism occurs in the matter of seconds, milliseconds, nanoseconds. It's a very short time. In our picture, we have a ligand-gated ion channel. On the left, it says right here, ligand-gated ion channel, which is why we're keeping the green, and the G protein coupled receptor on the right. The ligand-gated ion channel shows the workspace of individual subunit proteins, defining a pore where the flow of ions cross the cell membrane. Binding of the neurotransmitter occurs on the part of the protein located outside of the cell with this receptor, which is right here where this thing. Let me change the color back to red. This part here. Ligand binding domain. On the other hand, with the G protein coupled receptor, binding occurs outside the cell or in a so-called pocket that has formed by protein domains that spread across the membrane. So these are the pockets, which is also sort of like the conducting pore, but okay, it's a different story. And that's just a brief thing of neurotransmitters, which have to do with it, but aren't like the gist of it. That is all for the presentation. This is a list of our references in case you want to go deeper into each type of ion channel, or if you want to just see how neuronal communication occurs. You can do it here, or I am open to any questions if there is any to be asked. Thank you guys, and I'll see you on the next one.